In this tutorial, I am going to show you how you can identify variants in bacterial genomes using the tool SNIPPY. This is the page in case you want to read about the tool. I will leave the link to this page in the description box for you to use. What SNIPPY does is to help you to identify variants in haploid genomes. So it's not just bacterial genomes, but if you have genomes from haploid organisms, then you can use SNIPPY to identify variants in it. It was designed for that purpose actually, so that's why we are using it. So with SNIPPY, you can identify SNPs, you can also identify indels and some other variant types. SNIPPY generates a number of output files, and one of the files that I find very useful is the SNPs table. So with the SNPs table, you have this information here. It will help you to identify information such as whether your variants were found on chromosomes or plasmids, and then the positions and even the types of variants. These are important things. You can also identify the genes on which the variants were found, and then the products and even the variant effects. So what SNIPPY does is to combine a number of tools and then aggregates the result from these tools and then just put all of them in output files that you can use for your downstream analysis. So that's why SNIPPY is a cool tool. So with SNIPPY, we also need to specify a reference genome and then the input files, but we will talk more about this later on. SNIPPY can be installed with ease. You can use Anaconda or Brew. So these are things that you can do. For this tutorial, we will install using Anaconda. And so this leads me to the PC requirements. So in order for you to follow this tutorial, you need to make sure you have a PC that is running Linux or Mac OS. So it's important you get this. You also need to make sure you have installed Anaconda. We are going to use Anaconda to install a number of tools in addition to SNIPPY itself. So you need to make sure you have that. I have a tutorial that shows you how to install Anaconda. So I will leave the link to that video in the description box. So just use that. So with this tutorial, this is the workflow. We are first going to download some sequence reads. We will then trim the reads, and then we are going to call the variants. With the variant calling, SNIPPY requires a reference genome, so we are also going to make sure we download the reference genome and then use that. After that, we will do some post-VCF analysis. Okay, so this is just an overview of what we are going to do in this tutorial. So just stay tuned and let's proceed with the exercise. So now let's go. We will begin this exercise by first installing SNIPPY and some other tools. And therefore, we have to go to the terminal to do this. I assume that I have installed Anaconda, and so we will first add channels. We will add 2 of them. So we say conda config add channels. Let's add the first one, which is bioconda. Let's add the second one. So we say conda config add channels. And then we specify the name, which is conda forge. Let's list the channels to confirm that they have been added. So we say conda config show channels. This will list the channels we added, these 2. And we also have the default channel. This comes packaged with Anaconda, so we leave it as it is. The next thing to do is to create an environment and install the tools we need. Because there are a number of tools to install, I have prepared a YAML file that has the tools. So the YAML file has been uploaded to my GitHub repo, one of the repos, so we will go there. This is the repo. It's called the variant calling repo. So I will leave the link to this repo in the description box, so just use that. So I'll be populating this repo with tutorials, so you just try to monitor this channel. So let's proceed. So on this repo, we have the SNIPPY folder here. So just click it, and then you'll be sent to another page. So here you will see this environment.yaml file. This is what we need. And so let's first click it. You can also double click, and then the content will be displayed. So the name of the environment we will create is SNIPPY. We have some channels, and then the list of packages we want to install. And so to download, let's first click on the raw. Just come to the right side here. Just click on the raw button here. And then you have this being shown for you. So I'll show you two ways of downloading. So the first one is to just save it as a page. To do that, just right click. I'm using a file, so I'll just right click anywhere and then just go to save pages. And then I'll just save it. I can save it anywhere on my PC. Let me just use desktop just for demonstration purposes. So I have it done. So there's one way of saving it. The other way is to just copy this link here, this entire link here that I'm highlighting here. So copy it, and then you can also download this file via the command line. So that is what I'll do. I'll go to the command line and just download it from there. So another way is to also copy it here. Just go to the terminal and then just open a new text file and then just paste it there. But I'll just download it here. So let's go to the terminal and then proceed. So first of all, I'll just use wget to download. So I'll just type wget here and I'll just apply it with the link. So this will download the YAML file for us. So let's do this. You can also use any downloader that works on the command line. So I have it downloaded. Let me clear the screen. Do an ls and you'll see the YAML file there. So here you can just display it here just to see the content, but that's by the way. So let's clear the screen and just continue. So now that we have the YAML file, we will now create the environment and install. So the YAML file is much convenient because it prevents you from specifying all this list on the terminal. So let's see how it is done. So we say conda env create. We say -f and we specify the YAML file here. If you have downloaded it and it's not on your current directory, you just specify the correct path and just let conda have access to this file. So once you have this, you just run it. So let's run this to get our tools installed. And so with this particular command here to install, it might take some minutes. Okay, so I think I tried one and it took about 20 minutes. So just be patient whilst it loads. Okay, so just wait and then to install everything for you. So let's wait and see how it goes. Okay, so the tools have been installed. Let's first clear the screen and continue. The next thing to do is to activate the environment and test some of the tools. You will test just SNIPPY. So we say conda activates and we give the name of the environment, which is SNIPPY, and then we run it. Once the environment has been activated, you will have the name also being displayed on the terminal. So there we have it. Now let's just test SNIPPY. So we just say SNIPPY. And there we have it. So once you type SNIPPY and you execute it, you have this text being displayed. So this tells you how to use SNIPPY. You also have some information here, such as the outputs, the parameters, and then some inputs. So these things are there for you to just read and know how to use SNIPPY. So that's it. So now we have everything we need, and I'm assuming that you've also followed and gotten your environment activated. So we just know that everything is in place. So it's now time to start the practicals itself. So let's clear the screen and then continue. Okay, so now we have the tools working. And so the next thing we will do is to download the example data. I mean the sequence reads from which we will identify the variants. This data was generated by researchers who published this paper. So I have the paper here. So I will leave the link to this paper in the description box in case you want to read this paper. So with this paper, they sequence the Mycobacterium monstrans isolates. So they sequence the Illumina and then the nanopore reads. But for this tutorial, we will use the Illumina reads. So it has this accession number here. So that is what we are going to use. Okay, so with the paper, they combine the Illumina and the nanopore reads. But for this tutorial, because we just want to keep it simple, just for demonstration, I'll just use only the Illumina reads for demonstration purposes. So if you are interested, you can just join this. But I think we should just stick with just the Illumina reads. That's what I'll say. So I'll just download the Illumina reads. So it's a paired end sequence data. So that means we will have read 1 and then read 2. That is forward and then the reverse. So let's go and then just check it out. So the data can be found in the sra-database. So let's go there. So this is the link here. I'll leave this link in the description box in case you want to use it. This is the link to the page here. So when you are here, just look at the tabs here. Just click on data access. Data access here, and then you will see this tab here, okay, where you have this particular links here for the data. So the data has been stored in different servers. We will use the EBI one here. So we have the read 1 and the read 2. So let me just enlarge it. We will use this ones here. One for read 1, the other one is for read 2. So these are the links I will use to download the data. So let's go to the terminal and then just do it. But let me also say, in addition to using the terminal, you can just download using the browser. So if I click here, I'll be asked to save it. Okay, so you can use that to also download it, like what I have here. So you can use that to download both. But if you are working on a remote server where you have access to only the terminal, then you can just do everything straight from the terminal. So let's go to the terminal and then just download these files. So that's what we will do. Make sure you have copied the links. So let's go to the terminal. Okay, so we are going to use wget to download the data. So let's start with the read 1. So I'll just copy the link here again. Here, and I'll just come in and just paste it instead of just typing everything. So I'll say wget, I'll supply it with the link here. And then what I also want to do is, as I am downloading, I want to give the file a different name. So I'll say -o, and then I'll say raw_r1.fastq.gz. I am doing this because I will also process these reads. Okay, so these are raw reads, so I'll just process them. That's why I am giving them the prefix raw. And then the R1 indicates that this is the forward read. So take note of that. So let's run this. So it's a small file, so you can download it quickly onto your PC. So let's wait whilst it downloads. Okay, so we have the first data downloaded. So it has been saved for us. So do an ls and you will see that the file is there. Let's download the reverse read. So let's go and get a link. So this is a link. So let's copy it and then come and paste it also here. So I'll say I'll supply it with a link and then I'll give it a name, -o. I'll say raw_2.fastq.gz because this is the reverse read. So let's download this. Okay, so the reverse reads have also been downloaded. let's clear the screen, do an ls, and you will see it also here. So we are good to go. So let's clear the screen and continue. In addition to the sequence reads, we also need to get a reference genome of the isolate or the organism we are dealing with. If you take a look at the paper, we are dealing with a Mycobacterium monstrans isolate. With Mycobacterium monstrans, the reference genome is the AJY99 strain. This genome can be found on the NCBI database. So let's go to NCBI. This is the page. I will leave the link to this page in the description box, so just use that. I will give you two approaches that you can use to download the data. We are going to use a GenBank format. So let's look at how we do this. With the first approach, we will use the browser. So just follow my cursor, come to the right side. You will see this send to text here. So just beside it, there is this small arrow here. So just click it, and then you will have a drop down here. Make sure complete record is checked. When you come to choose destination, select file. When you come to format, make sure GenBank has been selected. If you don't see GenBank, click on it and select GenBank. After that, click on the create file button here, and then you'll be asked to save the file. So I'll save it on my PC. I'll also give it a name, AJY99.gb. That will be the name. So this is how we save it using the first approach. The second approach uses Python to do the download. So with Python, you will need to also have the accession ID. Okay, we need this. We also need a database, but I'll do the demo on the terminal, so just wait and see. So let's go to the terminal and then let's type Python. Python is one of the tools we installed. If you use the YAML file, then Python is also installed together with the required library. So let's type Python. If you have already downloaded a GenBank file, then you can skip the Python section. But if you want to learn, why not? Let's do it together. So let's proceed. So with Python, we will use the BioPython package. So we will import two models from it. So we say from bio import seq.io and then entrance. These are the two models we will use. Entrance here requires that we supply our email address. So I will say entrance.email equals, the email address should be in quotes. It's a string data type. So I'll say bioinformaticscoach at bioinfo.com. This is my email. It's an example. So I'll run this and then I have it there. Once we have this, we will now download the genome. So we will use entrance again. So we say handle equals entrance.efetch. We are fetching from the web. Database, which is DB, will be equals to new call. ID, also in quotes, it's CP000325.1. I'll leave this ID also in the description box in case you want to just copy and paste. Ret type will be GenBank, that's GB. Ret mode will be text. So this is what we do. So we have it there. And let me also say I have a separate tutorial that show you how to use Python to download sequences from the NCBI database. In that video, I cover a lot of stuff, so you can just watch that if you are interested to learn more about how to use Python for this kind of activity. Now let's run this. Let's run and then just download the sequence data. So we have it here. Okay, so once we have this, we need to also properly format it so that we can export. So we will say record equals seek.io.read. That will be handle. We specify the format, which is genbank GB, and then we run this. Okay, so let's move on to the next. So once we have this, we will now export this. So we'll say seek.io. Let's do it right. seek.io.write. We specify the sequence object, which is record. We specify the name of the output file, which is agy99.gb. And then we specify the format, which is genbank. And then we run this. So let's do this. Once the sequence has been exported and saved, you will have this appearing. This indicates that it was only one sequence that was saved. So that's what we have here. So now we are good to go. So let's quit Python. So there we have it. So if you do an ls now, you will see that you have the genbank file here. So these are the two ways you can use to get the reference genome. Now let's clear the screen and continue. So let's do an ls to check the files. So there we have them. We have our raw reads. We also have our reference genome. The next thing to do is to perform quality control, and we will do this by trimming the raw reads here. Trimming helps to remove adapters, helps to remove reads of low quality, etc. So trimming in general helps to improve the overall quality of your reads. And this is the same assumption we will use for this tutorial. We are going to do trimming. It is also important to inspect your reads. Okay, so inspection can be done using tools like fastq, multiqc, etc. There is also part of the quality control. fastq, for example, will do some analysis, generate plots and some visualizations that you can use to get to know the overall quality of your reads. So it is important you do this inspection. For this tutorial, I will skip the inspection aspect because I want to focus on the variant calling process itself. So just know that it is important to inspect your reads if you are going to do analysis on NGS and sequence data. So take note of that. Now let's get back to work. We are going to look at how we will do the trimming. Trimming will be done using the tool called Sickle. Sickle is part of the tools we installed. So if you use the YAML file here, then Sickle is already installed. So let's look at how to use Sickle for trimming. Let's type Sickle. So there we have it. With Sickle, you can run in the PE mode or SE mode. So if you are dealing with paired end reads, then you have to use the PE. If you are using single end reads, then you have to use the SE. Okay, so that's what you need to know. We are using paired end reads, and so we are going to use PE. So let's just type Sickle PE just to look at the usage. So we have some statements here. We'll just look at a few of them. So with Sickle, we specify the forward and then the reverse reads using this flags here, -f, -r. We also use -o and then -p to specify output files for the forward and the reverse trimmed reads respectively. So we will use these options here. With Sickle, we also need to specify the quality type. So with fastq files, the bases have qualities. Okay, they have quality scores. So there are different formats. Some use Solexa, some use Illumina, Sanga, etc. So we will use -t to specify the quality type. We are using Sanga, so that's what we will use. We are also going to use -s to specify an output file for reads without made pairs. Some reads may not have made pairs, so these are single end reads. So we are going to put them in a separate file. So that is what this flag here is for. Sickle can also be used to trim reads that fall below a particular threshold. When I say threshold, in terms of the quality. So -q here, when you specify, SQL will remove reads that have quality scores below the value that you set. -l is also used to trim reads based on length. So we will use these flags as well. And then we are also going to use -g here. -g here is used to zip the file so that it leads to compression of the files, and we can also save some space. So that is what we will do. So we are going to use these options for SQL. So let's clear the screen and just continue from here. Let's do an ls. Yeah, and now we can proceed. So let's SQL ourselves. So we say SQL p-f for forward read, so that will be raw R1, -r for reverse, that will be raw R2. Now the output forward trim reads, I will name it trim underscore R1.fastq.gz. Reverse will be trimmed underscore R2.fastq.gz with single end reads. I mean those generated from the trimming. We will say trimmed underscore s.fastq.gz. Notice the names I have here for the trim files. It is important to have naming conventions. So what I have here, if anyone comes to look at the files, the person will have an idea of what these files are because I have the name trim. So obviously this means that these reads have been trimmed. They are trimmed reads. Notice the names I gave for the reads, those we downloaded. I named them raw because we were going to do some processing on them, and that is why I named them this way. So these things helps you to know the files and even track the stages of your analysis. So it's important to have this as part of your plans. Okay, let's proceed. So now let's specify the quality. That will be Sanga. Let's specify the quality threshold. That will be 20. Let's specify length threshold. That will also be 20. So let's do that. So let's type them. Okay, so we have them here, and then let's specify -g. That'll be for gzipped. So now we are ready to trim. So let's do the trimming. So let's wait while SQL does the trimming for us. Okay, trimming has been done. So let's do an ls, and we will see that we have some names here. Okay, that's a trimmed. These are the trimmed reads. So these are the files we will use for the next phase of the exercise. So let's clear the screen and just continue. Okay, now let's do an ls just to check the files again. So we have our trimmed reads, which we are going to use for the next part of the exercise. So with the next activity, we are going to call the variant, and we are going to use SNIPPY to do it. Before we call the variant, let's look at how SNIPPY is used. So let's just type SNIPPY and then look at the help commands. So there we have it. So SNIPPY has lots of options that you can use. Some of them have got to do with input, some for resources, output, and we also have some parameters here. So we will use a few of these. So we will use some resources here. We will look at how we can use the CPU's resources here. So CPU resources helps you to specify a number of CPU cores that SNIPPY should use. The higher the number of CPU cores, the faster SNIPPY will run. That is assuming that the number you specified exists on your PC. You can also use RAM, you can also use this TMP there, but for this exercise, we are only going to use the CPU's resources. With the inputs, we will need a reference genome, so we will use this to specify we already have our GenBank file that we will use, but SNIPPY also accepts fastq and then EMBL. But this exercise will use the GenBank file so that we can also do some annotations. With SNIPPY, you can use input files of different formats. Okay, so you can use fastq files. So if it's fastq files, if they are paired, then you can use this option to specify the forward and reverse. If it's a single line, you can use this. Sometimes you may not have fastq files, but you may have the contigs. Maybe you have performed genome assembly and you have the contigs. You can also use that for the SNP calling or the variant calling. So for that one, you use this option to specify the contigs. You can also use paired and reads that are interleaved. If you have BAM files, you can also use this one here. Okay, so there are a number of files you can use here. So just look at the file format you have and check whether SNIPPY supports that. That's what I'll say. For this tutorial, we will look at how to use paired and reads and also how to use contigs. We will look at these two file formats. So let's proceed. So we also need to specify output files or output directories. So for this exercise, we are going to use the outdir option here. So when you specify this, SNIPPY will create a directory and then all files generated will be stored or saved in that directory. Alternatively, you can also use prefix. So that means that SNIPPY will generate the files or writes, and then the files will have the prefix that you specify here. So prefix has got to do with the name in some way. You can also generate reports and all these things, but we will just use this option here. With SNIPPY, you also have some parameters that you can just input. Okay, so one of the parameters can even help you to identify rare variants, but these are things that I will encourage you to explore. You skip this aspect here. So we'll just keep it simple in this exercise. So let's just go on and just do the variant calling. So let's clear the screen and then just proceed. We will just do an ls again and just look at what we have. Before we proceed, I made mention that with the fastq files, we will use forward and the reverse. But if you look at what we have here, we have forward, we have reverse, we also have single and reads. Single end reads may be useful in some cases, but generally we use the paired reads to do the analysis. That is if you've done trimming. I'm talking about single end reads that are generated after trimming. So that's what you need to know. So usually if you've done trimming, you have these ones, we can just use just this pair here to do the variant calling. But some activities like genome assembly, you can include both the pairs and then the single end reads as well. So take note of that. Okay, now let's get back to work. So we are going to use SNIPPY for the exercise, and so let's just look at how to use it. So to call the variant, we will say SNIPPY. Let's specify the CPUs. Let's use 8. Let's specify output directory. Let's use variants. Let's specify the reference genome. Let's use agy99.gb. That is our file. Let's look at the forward reads. So that will be what's trimmed underscore r1.fastq.gz. Let's look at the reverse reads. That will be r2. And that will be trimmed underscore r2.fastq.gz. Now we have everything we need. So let's run SNIPPY. Okay, so the process has started, so we will wait for SNIPPY to complete the variant calling. And please know that if you have lots of CPU cores, if you have more than 8, you can increase it, and this will also make SNIPPY run faster. So let's wait and see how it goes. Okay, so the variant calling process has been completed, and so you will have the statement here. So we are good to go. So let's clear the screen and just continue. Let's do an ls to check the files. So there we have them. You will find this directory also created. This directory contains the output files for SNIPPY. Let's take a peek into this directory. So we will say ls variant, and you will see the files listed for us. These three files, ref.fa, ref.fa.fai, and then reference, these three files are related to the reference genome, so I am going to skip them. We will look at descriptions for the other files. The descriptions can be found on the GitHub page for SNIPPY, so let's just go there, and we have them here. So it's the same link, so if you are here, you can just scroll down and then just come to the section where we have the output files. So we have the extensions and then their respective description. Let's check the terminal quickly. You will notice that the names of the files have the prefix snips in addition to their extensions like this one. So we have .bam, .bed, .tab, etc. So it is these files that have been given here. Okay, so let's look at them. So .tab gives a tab separated summary of all the variants. The same information can be found in a CSV format, which will be in the .csv file. The same information can also be found in the HTML file. We also have a VCF file that has the final annotated variant. We also have a bed file, GFF file. There are lots of files here, so depending on the information you want and the analysis you want to do, then you select the appropriate file and then just use that. Now back to the terminal. We will look at how we will explore some of these files. So let's just cd to the variants directory here. Now let's look at this one here, snips.txt. That is what we are going to start with. So let's do a cat. It's a text file, so just cat snips.txt should give us the information. So let's do a cat and we have them here. So here we have dates, read files, reference, reference size, software, and then we also have these ones. I will focus on these ones. So SNIPPY gives you the types of variants identified and also gives you their respective counts, and that is what we have here. And so the sum of these counts is what SNIPPY will give here. That's the variance total. So take note of that. It is also important to know that with SNIPPY, the initial variants that are identified are filtered. Okay, so the initial variants will all be found in this file, snips.raw.vcf. So SNIPPY does some filtering, and then the filtered ones, the ones SNIPPY identify, are used to populate the other files. For the filtering criteria, you may have to check the SNIPPY page to see if you can find them there. That is beyond the scope of this tutorial, so let's proceed. So with the files, some of them you can open with a spreadsheet software like the snips.tab and then the snips.csv. You can open with spreadsheet softwares. With the HTML file here, you can open with a browser. So let's look at these files. I'll just open them on my PC. Let's look at them here. So I think it's this ones here. Yeah. So let's start with the snips.tab. You can open with a spreadsheet software, LibreOffice, Microsoft, Excel, etc. So any of them should be fine. I think I am using WPS, so I'll just open them here. Okay, so I have the snips.tab here. So there's it. So we have some columns here, or headers, if you call it that way, with information. And then the rows here, the rest are the variants. So let's look at some of them. So the Chrome column here will give you which sequence, okay, that's the variants were identified from. So if, let's say, the reference genome had multiple sequences or multiple contigs, then this particular column here will tell you which of these sequences has that variant. That is what you need to know from this particular explanation. Okay, so here we are using just one sequence. Okay, so obviously they all have the same label, so it's fine. We also have the position and the variant type here. We also have effects, the variant effects. Okay, you need to understand the effect of the identified variants. So it's there. You also have the gene, and so with SNIPPY, because of the annotations, you can identify which genes have particular variants, or you can even look at the counts, maybe how many variants were identified on a gene. So these are information that you'll be able to get with SNIPPY. You also have the product, which gives some description about the gene. So just that. Okay, so the information here, we are able to get them, some of them, not all anyway. For some of the information, we are able to get them because we are using an annotated reference genome. Okay, so notice we used a GenBank format, so it has annotations, and that is what SNIPPY used to give us some of this information. If you use the reference genome in the fastq format, then some of the information may not be present, they may be absent, so take note of that. So that's fine. So that's for the .tab, the tab separated values. We can open the CSV file also with a spreadsheet software, the same as the tab. It has the same information, so that's what you need to know. So just look at them. With the HTML file, you can use your browser. Let's just open with a browser just to see. It's the same information. And so basically, SNIPPY gives you a number of options that you can use to open that particular summary. So that's what I'll say. So the same information, just scroll down, left, down, whatever. So that's it. So let's go back to the terminal. We are going to explore some of them here. Before I even go to the terminal, let's get back to the tab separated values. I'll use this as a demonstration just for you to know. So I think, yeah, this one here. Yeah, okay. I think it's already opened. I think it's already opened. Okay, yeah. So we have it here. So with this data here, if you're using a spreadsheet, you can even start some exploration here. And so let's say you want to get the variant types. You can just use the filter function here and then just use that to get the types. And you also get the counts. So you can do that with a spreadsheet software. You can also, let's say you also have, let's say you also want to look at the variant for a particular gene. You can do that also here. So if I use my filter function, I can do that. So let's say I want variant for DNA. It's already here. Just that one. Yeah, I can just filter and then just get only that one here. Okay, so I have it here. That's great. So we have them here. I think they are about 10 or so. Yeah, I think they are 10. So we have them here. Let's check. Yeah, okay. Let's check. Okay, I think I use DHAA. It's fine. Let's just change to DNA. Let's check. It should be somewhere here. Okay, we have a number of them. Yeah, this one here. We have DNA. Yeah, so there's one thing about using the spreadsheets. Okay, yeah, so there we have it. So we have that one here. So it's 10. Yeah, okay. Yeah, so notice I have to do a little bit of work and section. Okay, so that is what we are going to discuss next. So using the spreadsheet for some of this exploration may be cumbersome because you may have some errors. You may have to do some manual searching and clicking and dragging. Like what I was doing, I had it difficult trying to locate this DNA here because I had to just scroll through. We have lots of genes here. I had to scroll through manually, and then I even had to go and click one which was which had a different name, but I just missed it. So this is just a demonstration of how cumbersome it can be if you are using the spreadsheet just to do some of this analysis. And this brings me to the other aspect. So it will be much efficient, it will be much faster and much easier if you use tools that have been designed for it. Okay, so that's what we are going to. So programming environments like Python and R have libraries that you can use to manipulate files of this nature, like this CSV files and then the tab separated files. So we are going to do that next. Okay, so let's go to the terminal and then we'll do some cool stuff here. Okay, so it's very, very important. So let's do it. Before we even look at using Python for some of this exploration, let's just look at how we can use Linux to also do some quick survey. We already started with it by using the cat command here to display this. If let's say you want to get the number of variants, you have the VCF file here, you want to get the number of variants, then With some Linux commands, you can do it. So let's just do it. Let's clear the screen first, do an ls, and then let's proceed. So to count the number of variants in a VCF file, you can use this grep here, -vin-c. We specify our pattern here, and then just specify the name of the VCF file that you want. In this case, let's say the raw.vcf. Okay, this is the raw VCF, those that contain the initial variant identified. And so let's run this, and you get a number. We have 31,125. So it is these variants that were filtered. So that's how we use Linux commands to get a count for variants. By the way, I have covered a separate tutorial where I give you 4 different approaches to count variants in a VCF file using some Linux commands. So if you're interested, you can go watch that video. The link is in the description box. Now let's proceed. So I have the count here. This is for the raw. Now let's count the filtered, the ones SNIPPY identified or just selected. Let's put it that way. So we can use the same command grep. We are using this file here, and then we can just change the name. So this one is snips.fills.vcf. Let's run this. So we have what? 25,656. That's the number. So out of this, these were the ones selected by SNIPPY. Let's look at how we can also count the number of variants if the VCF file has been gzipped, like this one here. We use zgrep, but the options, the parameters we use are the same. So you take note of that. So let's do this. So let's run this, and you have the same number here. So that's how we do. So we've counted it. So notice we had the same number here and then here. Okay, so this is the total number of variants that was selected as the final ones. So that's how we do. So with some Linux commands, you'll be able to also do some exploration. So that's, by the way, if you come here with the snip.txt, since it's a text file, you can use your head snips.txt just to get it. So we have it here. You can use tail.txt, you also get it there. Let's clear the screen and continue. You can do the same for the tab file here. We can say, let's say head.tab. You can get some information here. So these are cool things you can do. Okay, with the VCF manipulation, there are tools that have been designed to do that. So tools like BCF tools, VCF tools, VCF filter, etc. can be used to manipulate VCF files. And I have started making tutorials on how to manipulate VCF files. I've started with BCF tools. And so if you're interested, I will leave that playlist at the end of this video, so you can just check it out and start some post VCF analysis. So we are just scratching the surface here with this kind of analysis. So let's proceed. Now, let's look at how we can use Python to explore the information that can be found here and then here. Okay, so you can use R, but I'll use Python to do this, and it will also be an opportunity for me to introduce you to some Python libraries. So let's call Python. Python is part of the tools we installed. So if you use the YAML file, then Python is already installed for you. So let's do this. So let's run it. So we have Python here. So we are good to go. So with Python, we can use some libraries here to open CSV files and then this file here. So one of the popular libraries is pandas. So that is what we are going to use to explore the data. So let's use pandas. We have to import it. So we say import pandas as PD. We will import it. So let's import it. So we are importing pandas, but we are also giving it another name, an alias. We are just giving a shortened form so that we use this for our work. So let's run this. So we have imported it. So we will now read the file. We will just use this. So I will say df equals pd here because it's pandas. We are using pandas here. But because we imported with this alias, we'll use the PD. So that's why I have pd.read CSV. And then let's specify the name. It's what? snips.csv. So we have it here. This is a comma separated value. So that's what the CSV means. Okay, so with pandas, you can just use this and then just read it. But if the delimiter is a different character like this one here, which is a tab character, then you have to specify. So if you were to use the snip.tab, then you have to use this here to open it. You will say sep equals /t. And this will tell pandas that this file here uses this tab to separate values. So that is what it means. But let's revert back to the CSV file. We will stick with that. So let's proceed. snips.csv. So that one, the default setting is the comma, so we are good to go. So let's run this. So now we have it done. So what we are going to do is to use this particular Python pandas library to answer some questions. We want to find the number of variants, the total number of variants. We also want to find the variant types. We also want to look at counting the various variant types. We want to count everything. And we also want to look at how we can extract information or subset information using the pandas library. So these are cool things you can do with Python. So with the df, this is data frame. So with this one here, what we have is that the arrangement will be columns and rows. That's what the data frame is about. So if you look at the one here, the one we open, let's just use this one here, which is enlarged. Okay. We have this one here. The first row here, that's the header. So that's the column names. Okay, so with pandas, it will just use the first row as the header. So that one is not part of the data. But the others here, these are rows. Those ones will be part of the data. So take note. So here, these are headers. So these are the columns. So Python will just give you that info. I think we have about 14 columns, but we will do everything on the Python terminal. So let's proceed. So this is just by the way. So once you have this, you can check the columns. So you can say df.columns. And you have the column names here. So that's it. So from here, you can decide to do some queries based on columns and others. But we are just scratching the surface. For pandas tutorials, I'll be making pandas tutorials and I'll upload them on my data science channel. So just go and check it out. So the channel link is also given in the description box. It's called Data Science Coach, but just check it out if you're interested. Now back to what we want to do. So the first question we want to answer is how many variants do we have? To do that, it's simple. We say df.shape. So we have it here. So .shape will give you the number of rows and number of columns. But the rows here are the variants. And so if you want that one, you just use df.shape. Just add your index. That means we want just this member here, the first member. So we have it here. So it's simple, just straightforward. So now we have the number of variants. Now let's look at the second one. If we want the variant types, not the count, but the types themselves, the types are in this column here. Okay, that's what we have here. Let's just look at the spreadsheet. That's what we have here. Okay, so here we just use the filter to get it counts, but we will do the same thing here in Python. So to get the variant types, we will say df, and then we just specify the column, and then we say not value counts, we say .unique. Yeah, this one here should do. So what this does is that it will get the information here, the info here, and then remove duplicates. So that's what it does. So let's run this. And so you will see that we have them here. We have 5 of them. Okay, so this is what it does. So it's very simple. So instead of just going to click and drag and all that, just one line of code will just do magic for you. So how do we do the count? We have the counts here. Let's look at this one here. So we have them here. How do we do it in Python? It's also very simple. So to do the count, we will say df, the same column, and this time we say dot value counts, and then we will count it. So this will return the count for us. So we have them here. So for each type, you will have the counts here. Okay, so this is how we do it. So this one also sorts it out for you, but those things, we just look at them at another time. So that's how we do the count. So it's very cool. Okay, so we've been able to do lots of stuff. So with Python, you'll be able to automate a number of these activities. You can even store the code somewhere and then reuse them. So just take it. Let's continue. Let's clear the screen. I'll just use this import os.system, and I'll just clear. So I'm just using this to run some bash commands. So, with bash, we can use this word to clear, but in Python, we have to use another way. So that's what I've done. Okay, so I have it cleared. Let's do a df.columns. Okay, so let's look at the next thing. We can subset, let's say you want to get a variant for the gene called DNA A. We can do that in Python. So we can say DNA A equals, and then we say df, we call df again, where gene equals DNA A. We have it here, and then we just close it. So let's run this. So once it's done, you can just count. So DNA.shape. Now we are experts, so we are good to go. So we have 10 variants. Okay, you can just display them. Let's say DNA.head. So with Python data frames, with Panda data frames, you can just use the head and tail as well, and get the first n rows, first 5 rows, last 5 rows, whatever. So that's what we are doing here. So we have them here. So that's how we do it. We can export this information to an output file. So that's what we are going to do next. So to save this subset, this extract here to a file, we can say DNA.CSV. And then just give a name. So let's say DNA variants.csv. And then we say index equals false. We have it here. So let's run this. Okay, so it has been done. So if you take a look at it, let's just open the one on the desktop just to see. If you take a look at it, you will see it here. I'm just trying to open it here for you. Yeah, you will see the file here. So let's open it. Yeah, so there we have it. Okay, so notice it was also stored in the variant directory because we had already seeded to that directory before starting the Python work. So that is why we do this. So now let's quit Python. Let's quit it, and then let's get back to our terminal. So let's do an ls, just to see things. Do an ls in variants. Yeah, okay, we are already in the variants directory. So we'll just look at it here. So you will see the DNA variants.csv here. Okay, so that's what I was talking about. Okay, so now let's clear the screen and then proceed with the next phase of the tutorial. So let's cd back to the previous directory. So let's say cd ... and then we are there. Let's check the input files, the ones we use for the variant calling. So ls should help us do it. With the input files, we used fastq files. We used paired and reads. But with microbial genomes, sometimes we may have our sequences as contigs. And so with SNIPPY, you can use contigs instead of fastq files to call variants. And so I'm going to show you how you can use contigs to also call variants. So let's proceed. I have an example data we will use. It's on one of my GitHub repos, and there's a page. I'll leave the link to this page in the description box. So we'll use this file here, the contigs.fastq. We'll use this file. So this file here was generated from a genome assembly activity, which I did. Okay, so that video, you can find that video in the description box. The link is there if you are interested. So I assembled think I used the raw routes or the trim routes. I think it should be the trim routes, but it doesn't matter anyway. So I just did the genome assembly to get the contigs. So that's all that you need to do. So I'll just download it. To download this contigs file here, click on it. You can also double click and you have this page. So just come to the right side, check the download button, just click it. And then after clicking, you have this page appearing, which will display the sequences. Okay, so once you have it, I'll just save this page. So I'm using Firefox, so I'll just right click and then I'll just go to Save Pages. You can also do it here. Come to this side, click on Save Pages, but I'll just use this here. So I'll save it on my desktop, and so I'm done. Another approach to get this contigs is to download using widgets or any command line tool for downloading stuff. So to do that, you need to copy the link here where my cursor is hovering on. Just copy the link and then go to the terminal. So let's do this. Let me also do a demo for this. So I'll use widgets and I'll supply it with the link and then I'll just execute it. So let's run this. So this will download for us. Now it's downloaded. So let's do an ls and you'll find it here. Let me do a quick exploration here. So I'll just say head contigs.fastq just to get it here. So I use this command here. So we are good to go. Now let's clear the screen. Let's do an ls just to check one more time. And now we are going to do the variant calling. So we are using SNIPPY. So for SNIPPY, we use the ctgs flag instead of the R1, R2. So we will say SNIPPY, let's use CPUs 8. Let's specify after this time, let's use variant 2. Okay, we already have this one here. So let's say variant 2. We need to specify the reference genome. So dash ref. It's what? AGY99.gb. So AGY99. And then you specify a contig using ctgs. You give the name. So now you run this command to call the variant. So variant calling has started. So we wait whilst SNIPPY does its work. So let's wait and see how it goes. So SNIPPY has done its job. So let's proceed. Let's do an ls and you find that directory also here. And so it's the same procedure we use for the other one. You can just do an ls, check the files, use Linux commands to count the number of variants, just explore these files here, use Python to get some information. So once you have this output file here, you just basically do the same thing we did with the other one here. Okay, so this is where I also highlight that if you have scripts that are able to process these ones, then you don't even need to modify them much. Just a little modification to specify the files here will give you the information you want instead of going back to just clicking, dropping, adding, and whatever. Okay, so this saves you a lot of time because you do less manual work. Okay, so with programming in general too, it's helpful to automate these stuffs. And so if you have multiple samples, then all the more reason why you even have to write your bash script, have your Python or R codes to help you process these things quickly. Okay, so this is how it is. So this brings me to the end of my tutorial today, and I believe it has been helpful. So if you have any suggestions on how to also improve these tutorials, I'll be very, very much happy to hear from you. So just put it in the comment section. Let's have some discussions and then go on. So if you're interested to learn how you can manipulate VCR files, please check this playlist and then just continue. Okay, so that'll be all for this tutorial. Thank you for watching and I'll see you in the next session. Goodbye.